not only is the climate changing rapidly, it is unprecedented in modern times. So the changes that we're seeing are really unprecedented in more than several 10,000s of years. And we know this with really, really great certainty at this point that there's virtually no possibility that the planet could be warming as much as it is without the influence and contributions of human contributed greenhouse gas emissions. And another way of depicting this, which many of you may have seen, is from Ed Hawkins' now famous warming stripes, which you see here. And so just to unpack these, what you're seeing is every year between 1850 and 2018 is depicted by a single vertical line. And they've used 1971 to 2000 as a reference point, so sort of a reference period sort of in here, and then colored the years in cool colors, blues, if that year was cooler than that baseline period, and in pinks and reds if that year was warmer than that baseline period. And so it should be pretty evident from the change in colors, what's happening. And I find it also useful just to reference, where we're at in it. So this is this year that I've put the arrow beside is when I was born in the late 70s, and we were still in this kind of cooling period. Relative to that baseline, at least, not overall, but relative to that. And then this is the early 2000s where a lot of grad students were probably born. So I like to think about the shifting baseline and the fact that, grad students today, depending on your age, but making some general assumption, were already born into this really, really warming planet and have seen these, have lived through these extraordinarily unprecedented times. And climate change, of course, is really ocean change as well, because the oceans absorb so much of the excess heat that we emit, as well as the CO2. And so it's probably not a surprise as well that the past several years in particular have been the warmest in the ocean as the ocean continues to take up all of that excess heat. So I started teaching an ocean climate change class in 2021, and this is the first year I presented, pointing out that 2020 was, at that point, the joint hottest year on record ever in the oceans. And what was really notable about that, so this is 2020, not sure if you can see my arrow. This is 2020 down here in the red bar, and it tied with 2016, which is really notable because 2016 is a year we're going to talk about a lot today because that was the year of the mega El Nino, the 2015-2016 El Nino, which is the largest El Nino ever on record. And so it's extraordinary that 2020, which was not an El Nino year, tied with the ocean warming that had been experienced during that massive El Nino. And then the warming just kept coming. So every year I've looked for these headlines, and indeed every year the ocean just gets hotter and hotter. So this was 2021, 2022, 2023. And then, of course, you'll probably recall 2023 ocean temperatures really just went right off the charts in a very worrying way. We had entered into this next El Nino phase. And then by early this year, the New York Times posted on the front page a headline that said scientists are freaking out about ocean temperature, which is not really something that we typically say about scientists, but I was definitely freaking out and feeling that. So I felt validated at least. So when we think about ocean climate change and climate change overall, we're often really focused on gradual change. But of course, the other thing that is making a lot of us wake up to climate change is extreme weather events. And there's no shortage of extreme weather events. These are unfolding every week, it seems, in places around the world, from hurricanes and massive floods. Out here where I live in British Columbia, BC has basically become one of the poster children for extreme weather events because we just seem to continually get hit with them. But this is the one that is still really resonant in our minds out here, the 2021 heat dome, in which temperatures broke Canada's all-time temperature record. The town of Lytton tragically burned to the ground, and hundreds of people died in that event. And it was a very extreme event that I think woke a lot of people up to climate change. Analogously, in the ocean, as I've already mentioned, El Nino is one extreme weather event. And El Nino, of course, is a natural oceanographic phenomenon, but it is one that we understand more and more is being intensified, becoming more frequent, and becoming longer, these events, with climate change. So it's becoming, El Ninos are becoming super fueled by climate change. And what you're seeing here is one way of visualizing El Nino. So what you're seeing is this El Nino really is a phenomenon of the Pacific Ocean, in which currents, basically everything gets reversed from east to west. And a sort of dominant signature of El Nino is this sweeping across from the Eastern Pacific across the equator of different sea surface heights. That's what you're visualizing here, but it ends up causing very different temperatures across different parts of the Pacific, and can actually become a global phenomenon, as it has with recent El Ninos. And so El Ninos are the dominant form of what is now known as marine heat waves. And what you're seeing here, circled in black, is the place that I'm going to talk about today and take you to today, Christmas Island, where I have worked for a couple of decades on coral reefs. So El Ninos are the dominant form of marine heat waves, which are now really a predominant signature of ocean climate change. And I'm just going to show you that in the past decade, there has been study after study documenting the link between marine heat waves and climate change, documenting that they have become longer and more frequent with climate change, that they are projected to increase under climate change, that they are having huge impacts already on global biodiversity and ecosystem services, that we can, with a lot of confidence at this point, attribute them to global climate change, and that they have been called the normal for the world's oceans. I don't really like that term, new normal, because I think we're in a phase that there's no rest point. Things are just going to continue to be very, very dynamic, with all of these things intensifying. And so as marine ecologists, we can think about how marine heat waves and other facets of ocean climate change are impacting the species and the ecosystems that we care about, from coral reefs and seagrasses to mangroves and kelp forests to rocky intertidal and mangroves and estuaries into the open ocean. All of these ecosystems are being affected. None of them is immune. But of course, some of them have greater vulnerabilities than others. And the latest IPCC report ranked marine ecosystems and their vulnerability to climate change. And not surprisingly, tropical coral reefs, warm water corals, which you're seeing over here on the left, are by far the most vulnerable to climate change, because they are so sensitive to small changes in temperature, which I'll unpack in a minute. But then the other thing to notice here is that in temperate waters, kelp forests are the most vulnerable. So it is not coincidental that my lab has decided to focus our research on climate change impacts on these two really key foundational ecosystems that have high vulnerability to the changes that we're seeing in our oceans. So I'm going to focus almost all of my time on coral reefs today, and tell you about the work that we've been doing there, focused on climate change over the past decade. So just to orient, as I'm sure you know, coral reefs are truly extraordinary ecosystems for a number of reasons. From a biodiversity perspective, they harbor an estimated quarter of all marine life, which is truly astounding when you consider that they cover less than 0.01% of the ocean surface. So this extremely disproportionate amount of marine biodiversity. And then we have also known for a very long time that reefs provide really important goods and services to millions, if not hundreds of millions of people around the planet. And of course, coral reefs, I think, often seem very, very distant from Canada, and probably very distant from Newfoundland. But they are found within the waters of 100 countries around the tropics and subtropics of the world. And many of those countries that they border are lower income countries. So countries that are disproportionately, in part because of this, feeling the impacts of climate change, but unjustly so in that they did not cause the emissions. This latest report, Rebuilding Coral Reefs, published in 2021 from the International Coral Reef Society, also estimates that coral reefs are worth over $1 trillion annually in various forms of people's livelihoods, fisheries, tourism, coastal protection, and other ecosystem services. So incredibly important ecosystems, both to biodiversity and to people. So why are coral reefs so vulnerable to climate change? So that really has to do with this very intimate partnership that they engage in. It's an obligate partnership that the coral animal has with a microscopic dinoflagellate that used to be called zooxanthellae and has gone also under all sorts of different taxonomic revisions and is now known by the mouthful name Symbiodiniaceae. And so these are single-celled dinoflagellates under a microscope, just look like these brown dots. And so it was really only with the advent of molecular sequencing techniques that biologists came to understand that there's not one type of symbiont inside coral animals, but potentially many, many different types, or there are many, many different types of symbionts out there, and different corals partner with different symbionts. So normally in a healthy coral under ambient temperatures, the coral tissue is packed with these symbionts. And then if the coral becomes stressed, it can be due to a number of different factors, but especially when they become stressed due to small temperature anomalies. And this can happen with temperature anomaly as little as 1 degree Celsius. The coral animal ejects the symbionts. At that point, the coral bleaches, and you've probably seen photos of ghostly white corals. The coral is not dead at that point, but it has lost, it's the breakdown of this partnership, and it's the symbionts that normally give corals their really vibrant colors. So corals at that point are typically kind of on a route to starvation. In previous bleaching events earlier on, those bleaching events were often rare, and when there are, or short-lived, I should say. And when that's the case, the corals can retake up their symbionts and recover. But if the temperature stress is prolonged and the corals actually starve, then it can lead to coral mortality. And so there's sort of a couple of different pathways here, depending on the intensity of the heat stress and the duration of the heat stress. So the other thing to note here is that it is still a really open question in coral reef science as to why certain corals bleach, why some end up on this path to mortality, why others recover. And there's an enormous amount of variability across different coral species, but there's also an enormous amount of variability even within the same species, and that can be across different locations, but even within the same location. So what you're seeing here is this Passalopera coral, 2 different colonies side by side, a photo I took on Christmas Island. So they're obviously being subjected to the exact same heat stress, but the one on the left has completely bleached and the one on the right is not. So this is just to highlight this enormous variability, and that in the field of coral reef biology, we are in some sense in a triage situation where we are desperately trying to figure out how to save these ecosystems. And there is a dominant line of thinking that figuring out how to enhance the heat tolerance of corals, how to get them through this century of warming, probably has a lot to do with these symbiotic relationships. So coming to the 2015-2016 El Nino, which I have already alluded to, I'm going to show you how that heat stress unfolded across the world, but I want you to particularly focus right on the center of this map. So across here, you've got the Hawaiian Islands, right in the sort of center north of the Pacific, and then traveling due south from Honolulu, just above the equator, is Christmas Island. So I'm going to take you from the beginning, January 2015, and those reds colors are the heat intensity. And you'll see that the heat stress landed on Christmas in about May of 2015. And it certainly was happening in other places as well. You note up north off the coast of Canada, we had the blob, that big heat stress. But unlike other areas, the heat stress landed on Christmas and just got a chokehold on it and didn't let go. And so I had been working on Christmas since 2009. And so we were out there well in advance of this heat stress event. And it turned out that out of all of the coral reefs in the entire world, Christmas Island and the one right next door to it, Jarvis, got hit the hardest in this El Nino, which remains the biggest El Nino on record. So that meant that we really had this extraordinary scientific opportunity to try to understand the factors leading to resilience of coral reefs under climate change. And just to show you another depiction of this, this was a tweet that NOAA, so the U.S. National Oceanic and Atmospheric Administration that runs Coral Reef Watch, sent out in the spring of 2016. This was for all of the Northern Line Islands. That's the island chain that Christmas is a part of. And the measure of heat stress that they use for corals is called degree heating weeks. And this is the y-axis going from 0 to 25 degree heating weeks. So corals can start to bleach at about 4 degree heating weeks. You can start to see mortality about 8. And at the time before this event, the basic understanding was that no reef on the planet would experience warming in excess of 25 degree heating weeks until about mid-century. And as you can see, the heat during that El Nino basically broke the charts and went off the charts. And so since then, due to warming there and in other places since, Coral Reef Watch has been having to rethink how they measure and quantify these heat waves, because we are getting hit with such intense heat waves that really exceed what had been expected even. So to take you to Christmas Island, as I said, it is due south of Honolulu, just a little bit above the equator. This is an aerial image of the atoll. It's the world's largest atoll by landmass, so about 150 kilometers in perimeter. It is part of the Republic of Kiribati, which is an island nation spread pretty much right across the Pacific in 32 very low-lying islands and coral atolls. And these are some of the people who live on Christmas, who we have been had the fortune to work with and interview for 15 years now about their well-being. Most of our focus, however, is as biologists underwater. And so what you're seeing here in all of these different colored circles are our 40 different monitoring sites, which are on the shallow fore reef of the atoll. So we work between 10 and 12 meters, scuba diving and monitoring these sites since 2009. And in part, the reason that I originally started to work on Christmas Island was because it really is in effect an ecosystem scale natural experiment, in that there is this very strong spatial gradient of local human disturbance. And that's because almost all of the people live up on this northwest coast. And then the disturbances to the coral reefs kind of fan out from there, minimizing by the time you get to the very remote ends of the island. And so to show you what this looks like, this is what the coral look like at our most highly disturbed site, which is right beside a very large pier. So there's been physical damage through dredging, lots of pollution, and barely any coral left at that most disturbed site. And then at the other end, one of our many very low disturbance sites prior to the El Nino, of course, but just showing extremely high coral cover, high coral biodiversity, just an extraordinary coral reef, extraordinarily healthy coral reef. And so those are the kind of the two endpoints, and then there's basically everything in between. So essentially, when we're thinking about ecological resilience, we can't study ecological resilience in static ecosystems. Ecosystems need to be stressed so that we can look at how they respond to that stress and see the impacts and if they're able to recover from them. So this really, this huge El Nino and this huge heat stress, which of course at the beginning we didn't know how bad it was going to get exactly. But we definitely understood it to be an extraordinary scientific opportunity. And so we really mobilized quickly with a plan to get out there. And I don't know if you can hear, but we're undergoing a fire alarm. So hopefully that is not too disruptive to you on your end. Hopefully you can't hear that too much. Very bad timing. Okay, so our plan was to try to understand which corals are resilient to climate change, to subject them to a huge stress test. And that stress test was this very intense El Nino caused marine heat wave. And then in addition to just monitoring the entire reef across all of those sites that I just showed you, we honed in on tagging individual corals, hundreds of them, and tracking them throughout the event to try to find out who the winners were, who the losers were, and really importantly, not just documenting what happened, but trying to get at the mechanisms of why some corals would perish and why some survived. So I want to acknowledge and really highlight the team here that I had the good fortune of working with. So science, and especially large scientific endeavors, of course, are not individual endeavors, but really team events. And so I had the good fortune of working with two amazing students, Danielle Klaar and Christina Tijan, as well as who really, took the lead on all of the coral tracking work. Other amazing graduate students, Jennifer Magel and Jamie McDevitt-Irwin, and then a suite of coral reef experts from around the world, because as if you know my earlier work, then you will know that I was trained as a fisheries scientist, not a molecular ecologist, and worked for a long time on fisheries impacts, but not climate change, not El Nino, and not coral reef symbioses. So at the time, this was all really new to me. And I was really, really fortunate to enlist and be able to work for many years with Ruth Gates, Kim Cobb, Melissa Guerin, Andrew Baker, and Ross Cunning, who helped to bring all of this work to fruition. So this is us out on our very small boat out on Christmas Island. I will say one of the things that is rather unique about working there, unlike where most coral reef research takes place around the world, is that there is no field station. And so we work in rather makeshift conditions, I would say, that we have managed to make work for a number of expeditions now. So this is us on our tiny boat, which is normally a boat chartered for recreational fishing. But you can see we've got all our dive gear packed at the end. And then this is the other end of the boat where we've got all the science gear and the whole rest of the team packed together. Lots of work takes place on the boat in terms of filtering samples in between diving, but most of our time during the day is definitely spent underwater, surveying the reef, using all sorts of different visual surveys, cameras, images, videos, and collecting samples. And then in the evenings, we are processing those samples right in our hotel room before falling into bed and sleeping, and then wake up and repeat for many days on end. So this is a makeshift water filtering station on the hotel room floor, and then that all gets cleaned up and put away, and then the whole thing repeats. So we were able to sample at the beginning of the event, and I'm just showing you here basically 2 months into the heat stress. And so I'm just going to take you through a series of photos to show you the progression of what it looked like on the reef. So this was 2 months in, and what we were seeing at that point was pretty highly variable bleaching. So some colonies were pretty heavily bleached at that point. Others were starting to bleach around the fringes, and other species had not even started to bleach at that point. Fast forward to December of 2015, and we were unfortunately not out there again until the following March. Just due to the cost of expeditions, we weren't able to get out there. But a recreational fisherman who was out there that we had been in contact with, got underwater, was diving while he was out there, and sent us these photos by email, just completely shocked at what he was seeing. And what he was seeing was reefs that in large part were completely bleached, this ghostly white coral that you see at the front. But even more worrying is what's all behind that, because what's behind that is coral that has already bleached, already died, and been completely overgrown by this filamentous turf algae. So while the corals at the front, have the possibility of recovering at this point, there is a lot of coral mortality already evident in this photo. By the end of March 2016, when we got out there, these reefs had been just sitting in this hot water bath for about 10 months, which is completely globally unprecedented, at least in modern scientific records, the amount of time people have been studying coral reefs. No one had seen a reef subjected to this much heat stress before. And so it was not surprising when we got out there that almost the entire reef was dead at that point. And so I'll just show you the juxtaposition of one of our very low disturbance coral reefs at one of those the very remote end of the island. This is before, or just kind of getting into the beginning of the El Nino, and you'll notice this very distinctly shaped coral up here, and then after 2 years later. So the same coral is still there. You can still see the structure, but it's dead, overgrown. And in this particular photo, there's essentially only 2 corals that are alive in the bottom right here, 2 of these mounding Porites corals that I'll come back to. So we crunched the numbers on this, having taken thousands of photos at each of our monitoring sites for many, many years before, during, and after the event, to quantify the impacts of this unprecedented heat stress on the reefs, and also to look at whether there were differences across the disturbance gradient. And so overall, we can boil these thousands of photographs and what we were able to quantify in them down to this one simple box plot showing the percent of hard coral cover and how it changed from before to after the event. And so overall, we documented that there was a loss of 89% of hard coral cover on the island. But you'll probably also notice that there's this huge error bars across this, and that has to do with the variability across the disturbance gradient. So prior to the El Nino, there was an enormous difference, as you already saw visually in the depiction of those 2 most extreme sites, this enormous difference in coral cover of the sites at the very low disturbance at the remote end of the island, spanning through to the sites at the very high disturbance end of the island. And so there was this huge gradient differences in coral cover that essentially just got completely flattened down during the event, with almost all sites ending at about 4% coral cover, give or take a little bit. The other thing that I want to point out is that in addition to mass coral mortality and mass mortalities that we're often seeing in marine heat waves, is that these changes are not uniform across species. And we're going to see this and are seeing this in every form of climate change, that some species, in some cases, not here, but in some cases, you end up having species that are actually responding positively to the changes and in others negatively. Here, everything responded negatively, but to very different levels. And so this is just to show you don't need to take in all of the different species names, but each color is a different species or genus of coral. And so just to show you that prior to the event, not only did the amount of coral really differ strongly across the disturbance gradient, but the community composition was actually really different. So there were a lot more of these blue-colored corals at the low disturbance end, and those are corals that have a competitive life history strategy versus these yellows and orange corals that were already dominant at the very high disturbance levels that have a stress-tolerant life history. And what you'll see is this big change in community composition at the very low disturbance sites, where almost all of the competitive corals perished in the event. And what was left were these stress tolerant corals. So this is one type of these competitive corals that you're seeing here. Just this enormous, this is a single coral colony with my grad student, my then graduate student, Danielle Clare, for size reference. These huge Acropora corals are also colloquially known as tabletop corals because they form these massive flat tabulate surfaces. And from what we could tell, at least at all of the sites that we surveyed, there was pretty much 100% loss of these corals during that event. And so another depiction of this changing community composition is thinking about the percent change in proportion of overall coral community. And so you've got some species that kind of increased their share of the real estate on the reef, and others like the Acropora that I just showed you here, and a couple other competitive species, Montipora, that really lost out on their share of the real estate. And so this is just to think about these winners and losers under climate change, which is, happening across ecosystems. So especially in the ocean, I would say, we're not just having mortalities from these extreme events and from other forms of climate change, but we're also seeing wholesale reorganization of marine communities. Okay, so there were some glimmers of hope, and I recognize that you have to really be an optimist to think that 11% survival is glass half full, but there were some glimmers of hope. So there were some corals that made it through the event. And I think it's important to point out here that coral animals, of course, are colonial. And so a single coral colony, like the one that you're seeing here, is actually made up of hundreds of individual coral polyp animals. And so one like this, where at the end of the event, it was, almost entirely bleached with these little patchy remnants, looked like it was on its way out. But in a lot of cases where the corals still had some of the polyps alive, they actually did manage to recover, like this one. In other cases, there were these branching Acropora, and overall, or sorry, not Acropora, Apocylopora. Overall, the Apocylopora did quite poorly. But again, it was highly variable, and some of them did just fine. So this one is at the end of the event. It's not even bleached, and it survived, continued to do well, and grow in the subsequent years. So I want to turn to think about specifically the corals that we tagged and tracked. What I showed you so far, those changes in the whole community, were from all of the photos that we took. But we also ended up tagging over 1,000 corals at sites spanning across the disturbance gradient. We took tissue samples from each of the corals that we tagged and did so at each time point that we revisited them, and then have done molecular sequencing specifically to look at the symbionts inside those corals. So here's the progression of the very first coral that we ever tagged. This is one of these mounding corals, Porites lobata, which is a stress-tolerant species and tended to do quite well throughout the event. Not this particular colony, but overall, this species had the highest survival out of any on the island. This is just a pairing of a couple of others that we tagged and followed throughout the event. Okay, so I want to take you through two different stories about mechanisms of resilience that we discovered through this tagged coral colony work. So the first was a real ecological surprise. And what I'm showing you here is the progression of this El Nino on Christmas Island where we were. And each of the dashed vertical lines here is one of the expeditions that we did to the island. And so you can see we've got this immediately before, early on, and then that kind of end towards the end of the event one in late March 2016. And so this is a story about Platygyra, which is also known as the brain coral, and how some Platygyra or some brain corals managed to survive this event. So one of the really dominant sort of lines of thinking in coral biology is that, as I said before, when corals become stressed by temperature anomalies, they eject their symbionts. And if the temperatures go back down to a level that is not stressful, then they can take up Symbionts again reestablish symbiosis and effectively recover from that bleaching state. But that can only happen, to our knowledge, if corals are not stressed. So here's what we ended up finding. You can probably see where this is heading. This is just one of these corals. We had tagged dozens and dozens of brain corals at sites around the island. This particular one, Coral 99, underwent this progression. So a couple of months into the heat stress, it was fully bleached, as we would expect. And this is, of course, the expectation. And then what we found was completely surprising in that we know that this coral had been bleached. Once we got there at the end of March 2016, we found it recovered while the water temperatures were still at stressful, elevated levels. So this, I just find this completely extraordinary in the sense that we never would have been able to figure this out in the lab. And I would say that the most common form of work on coral symbioses is to run controlled manipulative experiments in labs where people heat up the corals and see what happens. And so I love what we can learn by actually studying and watching nature really, really closely, because I think there are so many lessons that we can learn from studying organisms in real ecosystems that we simply can't learn from the lab. Okay, so how did this happen? And in order to understand that, we need to kind of peel back the hood and think a little bit about coral symbionts. So just very quickly, a high-level kind of simplistic overview of symbionts. This is the latest taxonomic revision, which has broken them into a number of different genera. And we can kind of contrast two of the most common types that we have on Christmas Island. Cladocopium, which used to be known as clade C, and I'll just refer to as the Cs, are often thought of as ideal symbionts in the sense that they transfer a lot of their energy, their photosynthate product, to the coral host. But like everything in biology, there are trade-offs. So they also tend to be quite sensitive to thermal stress. And so might be ideal under normal conditions, but potentially not so much under thermally stressful conditions. And that's where it may be more advantageous to have a partnership with Durastinium clade D symbionts, which are generally known as being heat-tolerant symbionts. So the trade-off here on the positive side, they're environmentally robust. They tend to deal quite well with thermal stress, but they also tend to be selfish, if we can anthropomorphize them for a moment, selfish symbiotic partners in that they don't share very much of their energy products with the coral host. So looking at brain corals that we sampled, these were the subset of sites where we sampled brain corals throughout this event. And what you're seeing are the different sites, and then on the right-hand side here, breaking down the symbiont community prior to the event. And so what you can see is that even prior to the event, there were these differences in the symbiotic community in that almost all of them, with these two exceptions, kind of clustered over here. And then the symbionts that were at the very high disturbance sites were quite distinct from the symbionts contained within brain corals at all different sites. And so these symbionts at the very high were all Ds. And that probably makes sense because they were already subjected to local stress in the form of local pollution and all of that other fishing pressure, dredging, etc. And the other ones at the other end, pretty much everywhere else, were Cs. What we found throughout the course of this event is that the type of symbiont contained within those brain corals did indeed dictate, or at least correlate, with which of those brain corals survived, except not in the manner that was expected. So what we actually found is that all of the corals that were at sites exposed to very high local disturbance prior to the event, and therefore already were in partnerships with D symbionts, all of them died, despite the fact that Ds are heat tolerant. And instead, almost all of the ones that survived were in partnerships with Cs. So this is really contrary to what was expected. So another ecological surprise here. This is just another depiction of this. And what you're seeing on the y-axis is the symbiont to host ratio, which is basically just giving you a measure of healthy corals having a quite high symbiont to host ratio, and then bleached corals having a low ratio. So what we found is that these corals bleached, those that started with D died. So we didn't, there's no way to track them further than that. The ones that started with C's bleached. And then at some point during the event, again, there's a bit of smoke and mirrors here since we weren't actually out during here. But what we do know is that during the event, when they were able to reestablish symbiosis while still subjected to heat stress, they did so with a different type of symbionts, specifically with those heat-tolerant D symbionts. And we know now that they have continued in that new relationship, that new partnership. This is just showing through 2017, but we actually know now, picking things back up in 2023, when we were able to get back out there after COVID, that they have persisted in those new partnerships. So some really interesting and quite unexpected pathways to survival here through even a globally unprecedented heat stress event, and also some evidence that underlying local disturbance actually matters, in that it was only the brain corals not exposed to very high disturbance prior to the event that actually made it through. Can I just check, do I just have until the hour, or is the time slot longer? I just want to check if I need to speed things up and skip over anything here.
You technically have, we have this room booked for another half an hour, but yeah, hours.
Okay.
So we have time for questions.
Yeah. Okay. I'm going to just skip through this next and maybe I might just skip ahead a little bit. And show you just quickly a little bit of work that students did, documenting the complete loss of soft corals, which is what you're seeing here. And then we also looked at the fish and found that there were huge losses in the fish right during the event. So the fish populations plummeted by about half during the event, but then rebounded within a year. And so what we suspect was going on is that the fish, being mobile species, were able to just move deeper, escape that worst of the heat stress, and then recover. Okay, I'm going to skip ahead to show you that there has been some coral recovery. And we had a 3-year hiatus unintentionally due to COVID, but we're able to get out back in 2023 and in 2024. And there has been this another El Nino over the past year. And really interestingly and positively, and maybe surprisingly, we didn't see any mortality in this latest El Nino. So that suggests that potentially that the most sensitive corals perished in the 2016 El Nino, and potentially that the reef is now made up of corals that are more stress tolerant and can withstand this heat stress. So some good news to end with for the corals. I'll just switch very quickly to say that during that 3-year hiatus, we also started working on kelp forests locally in British Columbia, and have documented marine heat wave impacts on kelps locally here. So I don't know if any of you have made it out to Vancouver Island, but we have the absolutely fantastic Bamfield Marine Sciences Center right out here on the west coast of the West Coast. And the body of water on which Bamfield is located is called Barkley Sound. And it turns out to be a very interesting place to study for a number of reasons. So first, this entire area of the Northeast Pacific was subject to this incredible heat wave that lasted even longer than the 2015-2016 El Nino, and became known as the blob out here because of essentially this huge blob of warm water that just sat in the region. But within Barkley Sound itself, which is what you're seeing here, there is this quite strong temperature gradient of about 3 degrees temperature. And so that allowed us to actually study how microclimate, how these small temperature differences can actually translate through to differential impacts during heat waves. So during this same heat wave period, in this case, 2014 to 2017, this was work led by my postdoc, Sam Starko. We documented really large losses of kelp forests in Barkley Sound. So what you're seeing up here, we had earlier data going back to 2007, and those are the photos in the top row. And there are lush kelp forests they're underwater, so you're seeing them from the surface, but lush kelp forests in this photo on the left and the right. And then afterwards, in 2020, when we were back out there sampling, those kelp are completely gone. Along the entire sound, so this is all of Barkley Sound, moving from this open ocean coast on the left through up the sound. This is about 16 kilometers, and this 3 degrees Celsius difference. And what you're seeing here are all of the different colors are segments where we assessed whether there was still kelp after this huge heat wave. And all of the dots in blue are areas where kelp persisted, and all of the dots in red are areas where the kelp vanished. And so probably not surprisingly in this case, all of the areas that were exposed to that warmer water further up in the sound disappeared. And indeed, overall, we estimate that about 40% of the areas have lost kelp. And we've been out there each summer since resurveying, and in general are not really seeing much recovery of the kelp. So big impacts on both corals and kelp. I'm going to wrap up. I'm going to spend 2 minutes moving to optimism and climate solutions. So all of this is pretty heavy, and I found it pretty heavy, and had spent about 2 decades documenting and diagnosing various problems in the ocean. And I think COVID was a good chance to take a step back and think about whether I wanted to continue to do that or focus more on the solutions. And so I'm going to just quickly show you 3 different initiatives that I have been heavily involved in building or leading over the past 4 years. So thinking about ocean climate solutions as a marine ecologist and ways that I can contribute, I've really shifted a lot of my research program from diagnosing problems to focusing on ocean climate solutions. And for us, that has meant switching to natural climate solutions, specifically blue carbon. And so we are leading a large Canada-wide initiative right now on blue carbon, trying to quantify the capacity for Canada's oceans on all coasts to serve as climate solutions. So the amount of carbon that is being sequestered and stored by Canada's tidal marshes, seagrasses, and kelp forests, as well as seabed sediments. And if anybody is working on that, I would be very happy to talk about it, or you can reach out by email. Secondly, here in British Columbia, I have helped to really build what is an initiative out of Banfield Marine Sciences Center called the Kelp Rescue Initiative, which is science-based kelp restoration. And so we are trying really to lead the charge on developing and testing different scientific methods for kelp restoration on the British Columbia coast. And then wanting to move beyond what I can do in my own lab as a biologist, I've, over the past couple of years, built a really broadly interdisciplinary training program, as was mentioned in the introduction, called Coastal Climate Solutions Leaders. And this is supported by NSERC Create and is in its 2nd year now, training students from right across the entire university who are doing their master's, PhD, or postdoctoral research. So they come together in very interdisciplinary groups and do coursework and professional training, development, and internships, all related to various aspects of climate change with a real focus on the skills that they need to join the climate action workforce. And I'll leave it there. I think I'm at time and happy to answer questions. Thank you.
I do want to ask if you could go back to the slide after the blue carbon slide, because I think your internet dropped for a second. Oh, or maybe again.
Oh. Is that so? There's that one. That one.
It dropped at this one, yeah.
Oh, okay. So you didn't hear any of that or see any of
Not
that?
really.
Okay, yeah. So, okay. So just to quickly say, Kelp Rescue Initiative is an initiative based at Bamfield Marine Sciences Center on the West Coast that I've been co-developing since 2021, and we are working on testing, developing, trialing different methods for kelp restoration in British Columbia.
Thank you.
Yeah.
We have time for questions. If anyone online has a question, if you could either raise your hand or just type it in the chat and then it'll get read out. But we'll start with questions in the room if there are any. Anybody got a question? Okay. All right. Let's see online. Tyler. You should be able to unmute now, Tyler, if you want.
Hello?
Hello.
Oh, you can hear me. Okay. Hi, Julia. It's Tyler Eddy. Thank you so much for the
Hi.
seminar. That was great to see all the work. I was curious about the switching between the different symbionts and if it is advantageous to have this. I think it was the D group that was the one that was surviving. Why do you have any theories or ideas why all the corals didn't have that symbiont as the default? And because it seemed like the other symbiont, if you started with D, you didn't do so well, if I understand. So do you have any theories about how these two things are playing out?
Yes, I do. And you've pinpointed it in that they remain theories because we haven't been able to actually do the analyses, and it's kind of been killing me for like almost a decade now. So I wish we could. But so there's a lot of evidence from much shorter term heat stress, whether in the ocean or in manipulative experiments, that Ds are advantageous. So often when corals start with D, they do much better than ones that start with C. The difference here was that not even so much that the intensity of heat stress was so great on Christmas. So temperatures were elevated by between 1 and 3 degrees Celsius, which is actually huge. But I think the bigger difference is the duration of time. So I think the difference gets back to that trade-off of how generous the symbionts are. So corals that are partnered with Cs normally are much better provisioned. And so they tend to also be in better health. Like they've built up larger energetic and lipid stores. And so I think when they bleached during this really prolonged heat wave, they had to endure months of being without their symbiotic partners. And I think it was the ones that started with Cs in this case that had those greater energy reserves to get them through the longer duration of heat stress. So that starting with Cs was actually an advantage in this case, whereas the ones that started with Ds maybe held on with their symbiotic partnership for a little while longer. Like they didn't bleach as soon because the Ds were more stress tolerant. But once they did bleach, which they eventually did just because it went on and on, then they didn't have many energy stores to get them through, and they ended up perishing. So I think it has to do with that trade-off. And this has just been heartbreaking to me in the sense that we took samples for lipid analysis, and we've just never been able to pull it off for a long variety of reasons. But I wish that we could, so that we could actually test it, because I think that's what happened.
Oh, that's really interesting. Thank you.
Yeah, thanks for coming.
Should be able to unmute now, Zintis.
For a great presentation, really interesting. I'm just curious about your blue carbon project. So out here at the Ocean Science Center, I'm currently working on using remote sensing to track kelp with some machine learning aspects. And I was wondering how you're sort of quantifying your coverage for that project.
Oh, great. That's so great to hear. Yeah, definitely. I mean, can get in touch afterwards.
Okay.
Email me. Yeah, we're doing lots of different things for kelp. We a lot of it is data that have been already collected. So we have a lot of data from Fisheries and Oceans Canada. We here on the West Coast, we ended up doing some work as well using a remotely operated vehicle, so at different parks. And then we actually, one of my postdocs, Jennifer McHenry, has an Atlantic kelp working group that met in Nova Scotia all the way back in the spring, I think, at this point.
Okay.
And I think she's, already compiled all of the data from various sources, but is still actively running the models. So she would probably really love to hear from you if you have
Right.
to share. But we have a pretty big working group from Eastern Canada and the Northeastern states on kelp. Yeah.
Okay. Cool. Thank you very much. I'll get in touch.
All right, we have a question in the room.
Yeah, hi. Well, congratulations on the presentation. It was really good. But I also had a question about the symbionts and if there are any corals, like I saw that they would transition from, I believe, like class C to class D, but is there any possibility for them to have both symbionts, like coexisting in a stable relationship? Or did you ever see anything like that? Or is it always switching from one to the other?
Yeah, great question. Within individual coral colonies, we almost always see that one of them is very, very, very dominant. So like way over 90%. So this is a great question. So there's sometimes can be very small levels of like a coral that is has C, but has a very, very tiny bit of like less than 1% of Ds in the background. And so there's definitely this question of shuffling or switching, like are they just shuffling the dominance of them, like ejecting the Cs, and then the Ds are just proliferating from within, or are they taking up the Ds from the environment? And there's lots of uncertainty about that. Yeah, in part because the sequencing techniques we're using aren't great for actually super clearly pinpointing the abundance. Yeah, so it's not common to have like a 50-50 type thing. We don't typically see that.
Thanks.
Yeah.
Any other questions? All right, well, thank you, Dr. Baum, for